We are now on the last group of fungal causing mycosis, and we refer to them as opportunist fungi that causes opportunistic mycosis. Opportunistic mycoses are fungal infections caused by saprophytes which under favorable conditions may cause diseases. Persons or individuals prone to this mycosis are those persons or patients on prolonged therapy with antibiotics or with immunosuppressive drugs. Those persons who have debilitating disease. Those persons with diabetes. And any person or any individual with immunologic deficiency. We have here the general characteristics between opportunistic fungi and pathogenic fungi. By knowing their general characteristics, we can identify the group. So for opportunistic fungi, they grow rapidly at 25 to 30 degrees centigrade on SDA or suborrowed dextrose agar. They grow poorly or not at all at 35 to 37 degree centigrade. They sporulate abundantly or sporulation is very abundant. They have a variety of pigmentation, meaning this group of fungal organisms are colorful because they produce many colors or many pigments. While the pathogenic fungi generally grow slowly at room temperature or at 37 degree centigrade. They need special media for sporulation and they do not display a wide variety of pigmentation just like the opportunistic fungi or saprophytes. The opportunistic fungi are the following. We have the Candida species. Of the four species of Candida, we have the Parapsilosis and Pseudotropicalis as opportunistic fungal organisms. Then we have the Aspergillus species, under which we have the Fumigatus, the Niger, Flavus, and Versicolor. Then we have the Zygomycetes group, which we have Absidia, Mucor, and Rhizopus. Still under the Zygomycetes group that are considered to be opportunists are Penicillium, Fusarium, Corvallaria, Acrimonium, Pacellomyces, Scopiolariopsis, and Verticillium. We'll not mention anymore the Candida species because they have been described in deep mycosis too. So we'll go directly to the Aspergillus species. The Aspergillus are opportunistic invaders infecting various sites of the body causing aspergillosis. The infection may be in the form of invasive infection, colonization, toxicosis, and allergy production or formation. The species is found widespread in the environment and commonly found or identified as a common laboratory contaminant, especially in different cultures. The rate of growth usually rapid, mature within 3 days. The colonial morphology, the surface of the colony at first will be white in color, then it will change to any shades of yellow, green, brown, black, depending on species. The Aspergillus species have septate hyphae or hyphal structures. They also have an unbranched conidiophore which arises from a specialized structure we call the foot cell. The conidiophore is enlarged at the tip, forming swollen vesicle. Vesicles could be completely or partially covered with flask-shaped phyalides, which may develop directly on the surface of the vesicle, giving us the uniseriate form or uniseriate phyalides. Or these vesicles are supported by the medulla or secondary branch of phyalides, giving us the biseriate form or the biseriate phyalides. Phyalide produce chains of mostly round or sometimes rough conidia 2 to 5 micrometer in diameter. For the Aspergillus species, it is best identified through their microscopic morphology or characteristics. So what are the different factors? We have the conidiform, whether it is short or long or slender, whether it is smooth or rough. So we have the term echinolate, meaning conidiform with thorns. Then we can also identify them by their vesicle, whether it is round or ovoid or small or large. And we can also identify them by their phyalide, whether uniseriate form or biseriate form. And lastly, we can also identify and differentiate the species by the arrangement or direction of the conidia or microconidia on conidial heads, whether it is parallel, irregular, or partial, radiation, or arrangement, or full radiation, or full sunflower arrangement. We have here the different structures of an Aspergillus species. We have the conidiophore, we have the vesicle, the medulla or medullae, then we have the conidia or microconidia. So again, on the lower illustration, we have the conidiophore, the phyalide, the vesicle, and the conidia or microconidia. So on the left illustration, no, we have the conidiophore, and at the tip we have the conidia. Now on the lower portion of the conidiophore is a branching of the conidiophore, which we call the foot cell. So the foot cell is actually part of the hyphal structure of the organism. Then we have here the flask-shaped structure we call the phyalide. So, which the secondary branch of the flask-shaped structure is what we call as the medulla. Then on top of this secondary branch is another layer of flask-shaped structure. In some source, it is referred to as stigmata. Or we have part of the conidia formation, which will later on lead to microconidia formation, or a chain-like formation of smaller conidia. That's why it's referred to as microconidia. So again, the terms that you should be familiar with are the conidiophore, the vesicle, the phyalides, and the conidia or microconidia formation of the species or of the organism. Phyalides are flask-shaped structures on the surface of a vesicle. The uniseriate phyalide, the cells or the flask-shaped structures are borne directly on the surface of the vesicle, surrounding it as a layer, so there will be only one layer observed. For the biseriate phyalide, the first layer is supported by another layer of phyalides. We call the secondary branch or medulla. So in biseriate phyalides, usually there are two layers that are observed, and on the first layer or surface layer are the conidia formation. We have here an illustration of uniseriate and biseriate phyalides. So we have here the conidiophore, the vesicle, and on the surface of the vesicle, if there is only one layer that are observed, then we have the conidia or microconidia formation. On this first layer, we have the uniseriate phyalide. Now, on the left side, we have two layers of phyalides that are found on the surface of the vesicle. So we have the first layer, then there is another layer of phyalides wherein on this first layer, the conidia formation can be observed. So this is a biseriate phyalide. We have here the conidial heads of the species, which is also used to differentiate them. So on the left side of the illustration is a regular arrangement of the microconidia or conidia on the vesicle. So if there is full arrangement of the phyalides and the microconidia, if it fully radiates, covering the entire vesicle, it is a full radiation or a sunflower-like formation of the conidial heads. Then on the right side, we have an irregular arrangement of the microconidia that are found on the surface of the vesicle. So we have a regular arrangement, we have an irregular arrangement leading to a full radiation of the phyalides and the microconidia of the species. Then in the lower portion is also the radiation of the microconidia, which partially covers the vesicle. So we have this type of conidia wherein there is partial covering of the vesicle. So we will compare this to the upper middle illustration. So in the upper, it's a full radiation or a sunflower arrangement. And in the lower illustration, it is just partial. So this is another point in differentiating the different species. And on the left side, lower portion of the illustration are the arrangement of the phyalides plus the microconidia, which will be considered as parallel to the conidiophore. For the conidiophore, on the left side of the illustration is a short conidiophore, while on the right side we have long and slender conidiophore. Then in the lower illustration, we have a rough conidiophore and we have a smooth conidiophore. So the appearance of the conidiophore is also used to differentiate the different species. The first species is the Aspergillus fumigatus. It is the most common cause of aspergillosis. This species is a saprophyte with worldwide distribution and commonly found in house dust. It occurs in outdoor and indoor air, in different types of soil, and on decaying plant material. It could also occur in compost, in wood chips, in feathers and bird droppings, also in self-heated hay and crops. This species is an important causative agent of systemic mycosis in domestic animals and in humans, especially those with immunocompromised individual or immunocompromised patients. Infection of this species are seldom acute, but the cardiac and urinary system as well as the brain may be attacked. Occasionally can also be found in human ear and eye infection. The morphology of all species of Aspergillus will be at first white on the surface, then it changes to any shades of yellow, green, brown, black, depending on the species. The reverse side of the culture medium or culture media will be white to tan in color. The microscopic characteristics of this species is that they have short conidiophore, less than 300 micrometer in length, and the conidiophore is smooth. They also have uniseriate phyalides that runs parallel to the axis of the conidiophore. We have here an illustration of the microscopic appearance of the fumigatus. So take note of the arrangement of the phyalides and the length of the conidiophore. So we can say that the phyalides are in parallel with the conidiophore. The next species is Aspergillus flavus. Aspergillus flavus produces the carcinogenic mycotoxin we call aflatoxin, which is a very toxic substance which often contaminates food such as grains and nuts. After Aspergillus fumigatus, Aspergillus flavus is the second most Aspergillus mold infecting human, hence the second most common pathogen of aspergillosis. Aspergillus flavus invades blood vessels in human lung or brain and cause infarction. Aspergillus flavus grows on moldy corn and peanuts. Warm soil, foods, and dairy products are other sources of growth. It can also be found in water-damaged carpets and in building areas. Aspergillus flavus has been reported to be allergenic, and its presence is associated with asthma. It has also been associated with aspergillosis of the lungs or disseminated aspergillosis, and also involved in ear and eye infection. Infection of the lung, the heart, and bladder have also been reported. Some strains of Aspergillus flavus are capable of producing a group of mycotoxins in the aflatoxin group. These toxins are carcinogenic and have been linked to a wide variety of human health problems. The surface colony of Aspergillus flavus is yellow-green in color and gold or red-brown underneath. On Zapic duck's agar, the colonies are granular, flat, often with radial grooves, yellow at first but quickly becoming bright to dark yellow-green with age. Conidial heads of flavus typically radiate, mostly 300 to 400 micrometer in diameter, later splitting to form loose columns, giving us the biseriate form, but having some heads with phyalides borne directly on the vesicle. The conidiophores are hyaline and coarsely roughened, the roughness often being more noticeable near the vesicle. To give a summary of the microscopic appearance of flavus, the conidia are globose to subglobose, around 3 to 6 micrometer in diameter and pale green in color. It gives us a biseriate phyalides. Conidiophore is conspicuously echinolated or rough conidiophore. Some strains produce brownish sclerotia. An illustration of peanuts and corns which are contaminated with Aspergillus flavus. The third species is the Aspergillus niger. This is the most abundant species of Aspergillus in nature, and it can grow on a great variety of substances or substrates. It can even grow in environments with very little nutrients available and is often found growing on damp walls in houses. This is considered to be the third most often cause of aspergillosis. As mentioned, Aspergillus niger could grow or found in and upon the greatest variety of substrates, including textiles, grains, fruits, vegetables, and soils. Commonly associated with fungus boll, a condition wherein fungus actively grows in the human lung forming a ball without invading the lung tissue. Aspergillus niger have been reported also to cause skin diseases and are a common cause of fungal-related ear infection we call otomycosis infection. And it is also considered to be a laboratory contaminant in different cultures. It can also cause mycosis in debilitated persons. The colony of the species is woolly. At first, it is white in color, then changes to yellow to black. So on the surface of the colony is black color, and on the reverse side of the culture medium or media is white or yellow in color. So we have here illustrations of the colony of the species in different culture media. So in the upper we have the Saborodextrose agar, and in the lower we have the potato dextrose agar. So common to both is the black color of the surface. The microscopic appearance of the species have long and smooth conidiofores with biseriate phyalides. The phyalides have a complete radiation around the vesicle, giving us the sunflower arrangement. The fourth species is the Aspergillus versicolor. This species can be found in temperate climates, in air and house dust. It can indicate signs of moisture problems in buildings and can be found in water-damaged building materials. This species produces a mycotoxin we call stigmatocystine, which is toxic and carcinogenic. The species has a characteristic musty, earthy odor, often connected with moldy houses, and is the cause of eye, nose, and throat irritation. Colonies in potato dextrose agar may range in color, pale green to greenish beige to pinkish green to salmon green or dark green. The reverse side of the culture medium will be reddish or uncolored in appearance. Growth rate is slow to moderate. Microscopic appearance of the species, they have hyphae that are septate and hyaline. Conidial heads are biseriate and loosely radiate. Conidiophores measure 120 to 700 micrometer in length, are hyaline to pale brown in appearance, smooth-walled and brittle. It can give us an appearance that it can be snapped off. Vesicles are small, approximately 9 to 16 micrometer in diameter and variably shaped, with medullae and phyalides covering most of the vesicle. The microscopic appearance of the conidial heads of the species. The main diseases of aspergillosis are allergic bronchopulmonary aspergillosis, acute invasive aspergillosis, and disseminated invasive aspergillosis. These diseases can be intermingled. The allergic bronchopulmonary aspergillosis, or ABPA, is a disease where a person's immune system is hypersensitive to Aspergillus spores. This hypersensitivity causes allergic reactions in the persons when the exposed person tries to expel the spores from the body. People with cystic fibrosis or asthma are especially vulnerable to allergic bronchopulmonary aspergillosis, with approximately 5% of asthmatics suffering this disease at some point in their life. This ABPA is usually caused by Aspergillus fumigatus. Tests for allergic bronchopulmonary aspergillosis can be performed through X-rays, skin tests, or blood tests. The chronic pulmonary aspergillosis and aspergilliomas is characterized by the formation of fungal ball in the lungs. The fungal ball, which is referred to as aspergilloma, makes the person sick because of the allergens or toxins it puts out into the person's body. Aspergilliomas can be detected through X-rays or blood tests. In some cases, surgery might even have to be performed to remove the aspergilloma from the lungs. We have another group of opportunists under the zygomycetes group. We have Obsidia, Mucor, and Rhizopus. For this group of opportunists, we have to be familiar with the following terms. We have stolon, which refers to hyphal structure, and stolons of this group is described to be xenocytic. There are no walls or there are no division. We have the term sporangiophore. We have columella, which means shoulder or collarette. We have sporangium, sporangiospores, and we have the roots, which are referred to as rhizoids. We have here the characteristic structures of the fungal organisms. So we have the roots, which are the rhizoids, and the hyphal structure referred to as the stolon, and in the stolon arises the sporangiophore. And at the tip of the sporangiophore are the vesicle, wherein the vesicle is covered by a ball-like structure we call sporangium. Inside the sporangium are sporangiospores. And if the sporangium is damaged, so it will be taken from the vesicle, and some of the species will actually have a columella or shoulder left on the vesicle. So take note of these structures because this will help us in identifying the species. The term zygomycetes is not the name of a particular fungus or fungal organism, but rather the name of a class of fungi. This class includes three orders. We have Mucorales, Morchirellialis, and Entomophthoralis. These orders in turn contain a number of specific fungal organism or specific fungi. The colonies of Zygomycetes is coarse, woolly, gray surface that covers the agar with fluff resembling gray cotton candy. The rate of growth is rapid, mature in 4 days, and the reverse side of the culture medium is either gray or white. This characteristics is common to all species of zygomycetes. So in the next slide, we can see the appearance of the colony, and this is, take note, common to all species. We have here the surface appearance of the colonies of Zygomyces species, and this characteristic is common to all species. The next group is the Obsidia species. The Obsidia are ubiquitous in nature, so they are found everywhere and is a common contaminant in culture in the laboratory. This is an etiologic agent of psychomycosis in which invasion is always preceded by breakdown of the normal body defenses. So mycosis in this species is often systemic, with invasion of the central nervous system, the arteries, the lungs, the GIT, and other parts of the body. May also be localized in subcutaneous tissue. The Obsidia species have a wide non-septate hyphae. Sporangiophore arise at the point on the stolon that is between the rhizoids. Take note of these characteristics because it will also help us identify Obsidia from the rest of the opportunists. Sporangiophore are branched and widen just below the columella. The columella, a typical shape like semicircle with a small projection on top of the vesicle. Sporangia are slightly pear-shaped. When a sporangial wall is destroyed, a short collarette often remains where the wall met the sporangiophore. So this is the columella or collarette. This is an illustration of the microscopic appearance of Obsidia species. Take note the most important characteristics in order to help us identify the species from the rest. Take note of the rhizoids or the roots. The sporangiophore arises in between these rhizoids. The next group is the mucor species. The mucor species have a wide, practically non-septate hyphal structure, and the sporangiophores are long, often branched, and bear terminal round spore-filled sporangia. When the sporangium disintegrates, fragments, round or slightly oblong spores are scattered, revealing the columella and sometimes leaving a collarette at the base of the sporangio. Take note, in this species, there is an absence of rhizoids. The last group is the rhizopus species. The rhizopus species have broad hyphae or hyphal structure with no or few septa. Numerous stolons run among the mycelia, connecting groups of long sporangiophores that are usually unbranched. At point where the stolons and sporangiophores meet, root-like hypha or rhizoids are produced. So take note, in the illustration, we have the rhizoid, and from the rhizoids will arise the sporangiophores. Sporangiophores of this species are long and terminates with a dark sporangium containing a columella and many oval, colorless, or brown spores. No colorate remains when the sporangule wall fragments or disintegrate. The rhizopus species can be differentiated from the other groups by the presence of stolon rhizoids and usually unbranched sporangiophores, and it is differentiated from Obsidia by the location of the rhizoids in relation to the sporangiophores and by the shape of the sporangia. So that ends up the first group of zygomycetes. Then we have another group of zygomycetes we call other opportunists, and that is our next topic and the next set of PowerPoints.